in your MBBS first year on the isomerism. Not many questions there, here and there 1 or 2 questions. And in carbohydrate metabolism, from the theory point of view, you will get a question. You can get a question on small notes like hereditary fructose intolerance, classical galactosemia. In long notes, 2 major questions, you can write them. One is the hormonal regulation of blood glucose, that usually comes as a long 10 marks question. Now what they do is they'll ask it in a clinical way, because nowadays they want to ask some clinical question also. So one of those questions can be asked in the clinical ways, hormonal regulation of blood glucose, in which 1 3 marks or 4 marks or 5 marks question can be one of the hormonal, write down the reciprocal regulation of glycolysis and gluconeogenesis. So all of that is covered under carbohydrate. Then glycogen storage diseases, you can get a short note. We expect you to write at least, when you are writing, type 1, type 2, type 3, type 4, type 5, type 6. Even if you write till Hirsch's disease, then it is fine. Then even if you miss on 7 or 8, usually the marks are not deducted, provided how good you have written the first 6 disorders. So glycogen storage diseases, that is a very important question. Also from the NEET point of view, from the entrance point of view, glycogen storage diseases is the most important question. And you might find out that at various times we have seen crossovers, like insulin is regulating glycolysis and glucagon is regulating the same process, but the reverse of it rather, and in the reverse direction it becomes gluconeogenesis. So when you see these kind of questions, well-fed, starvation, what is insulin, glucagon doing, it takes time to develop. So do not get irritated that I have just studied carbohydrate, there is too much of regulation about insulin and glucagon, because by the end, once you have covered in physiology, endocrinology, where they have taught you in detail insulin, once you have covered in the biochemistry, lipid metabolism, protein metabolism, all these things comes little easier. For the start on the go, it looks little difficult, but in the end, after your second, third semester is over, so by the time you have that final examination, it will be all will be clear, because I don't know when are they teaching physiology, endocrinology and all. So usually it is integration topics. Few topics are taught in biochemistry, few topics in physiology, and by the end, once it is all over, then there becomes a clear picture. So initially, whatever is pertaining only to carbohydrate, try and understand only that. So make sure that you have revised carbohydrate from the notes. Make sure you have taken the class note seriously, revise it, and then solve the questions. So just to give you some insight about carbohydrate, let's first do 25 questions, and then later complete notes is always advisable for the knowing this complete topic, right? So I have already told you the short answer and long essay type questions which can come from carbohydrate. Let's also see some MCQs. Now, question number 1. Which of the following is activator of gluconeogenesis but not its substrate? So which of the following is activator, meaning it will activate but is not a substrate for gluconeogenesis. It will activate the process, and that is seen with acetyl-CoA. I'm sure under gluconeogenesis, you might have encountered 4 enzymes. So gluconeogenesis, there are 4 major enzymes. We have number 1, pyruvate carboxylase. You are adding carbon dioxide, the name of the enzyme will be carboxylase. And then we have PepsiK, phosphoenol pyruvate carboxykinase. Then the third enzyme is a phosphatase, fructose 1,6 bisphosphatase. and the last is the glucose 6-phosphatase. This enzyme here, pyruvate carboxylase, this enzyme here converts pyruvate to, it's carboxylase. So it will add carbon dioxide to the pyruvate. It will become, from a 3-carbon compound, it will become 4-carbon oxaloacetate. And this reaction requires an obligate activator, and that is acetyl-CoA. Obligate activator means it is must required for the activity. Acetyl-CoA, although never will form glucose, acetyl-CoA never forms glucose. It is formed as an end product of glucose metabolism, but it never resynthesizes glucose. Then this acetyl-CoA never forms glucose, but it is must required for gluconeogenesis. And gluconeogenesis occurs only and only in liver. The site for gluconeogenesis is only and only liver. The substrate for gluconeogenesis can be number 1, lactate. Lactate can be the substrate for the gluconeogenesis. Number 2, odd chain fatty acid. Number 3, glycerol. Number 4, gluconeogenic amino acids. The most important source is gluconeogenic amino acids. If you have any doubt anywhere in carbohydrate metabolism, please put it in the chat box. We can discuss that also in today's session. So lactate, odd chain fatty acid, glycerol, gluconeogenic amino acids are the substrate for gluconeogenesis, but never a substrate for glucose is acetyl-CoA. So gluconeogenesis is never formed from acetyl-CoA. If the most important substrate for gluconeogenesis is asked, then it is gluconeogenic amino acids. Out of 20 amino acids except leucine, all are gluconeogenic. Then let's see another question. Which of the following best indicates the elevated blood glucose over a period of weeks? Now here, ketone bodies have nothing to do with the blood glucose. Hyperglycemia can be an indicator of diabetes. So it is elevated blood glucose will not remain for weeks. Even in diabetics, there will be change in the blood glucose levels depending upon the fasting and starvation. However, this looks like that it will always be high in diabetes, but it will be higher than the normal range, but it is not the best indicator, because blood glucose will keep on changing depending upon the well-fed and starvation condition. So it is an indicator but not as the best indicator. Lipids do not tell us about the elevated blood glucose. So the best parameter is HbA1c. HbA1c is when circulating glucose is high, it combines with hemoglobin to form HbA1c. To form HbA1c. HbA1c will give us the blood glucose control of over 2 to 3 months. For over 2 to 3 months, because the RBC lifespan is around 120 days. So on an average, you will get a blood glucose control of over 2 to 3 months. And the cutoff for HbA1c is 6.5 gram%. So if you get a blood glucose of more than HbA1c of more than 6.5 gram%, then it is diabetes. So the problem with blood glucose is this only. When you are diagnosing a patient, if I get a blood glucose report of 150 milligram per deciliter, it is inconclusive. I don't know whether the patient was fasting. Even if it is said that it is fasting sample, I don't know that what were the conditions on the over the last 3 days, whether he has taken steroids or not, because there are many medications which tend to increase blood glucose levels. So you have to have either 2 consecutive readings of a well prepared sample. A well prepared means you have to ask the patient to have a normal diet over the last 3 days and have a fasting of 8 to 10 hours, then give a fasting blood glucose sample. Or you can also take random sample, but then it has to be on 2 consecutive days. That is the problem with blood glucose alone as a measurement of the indices for elevated blood glucose. Then, which of the following enzyme deficiency is responsible for classical galactosemia? Let's quickly see galactose metabolism. You already must be knowing this. This is the last topic in our notes. Galactose is converted to galactose 1-phosphate. We are adding phosphate. Whenever you add phosphate, the name of the enzyme is kinase. So this enzyme is called galactokinase. Depending upon where it is adding phosphate, we call it kinases. Like fructokinase, it is adding phosphate on fructose, glucokinase on glucose. So galactokinase will add galactose, pay phosphate, and the donor of this phosphate is always ATP. Now galactose 1-phosphate is converted to glucose 1-phosphate. Simultaneously, UDP glucose is converted to UDP galactose, and this enzyme here is galactose 1-phosphate uridyl transferase. I am not rewriting it, it's written here only, and some authors call it GALT, others call it GAL1 put, the shorthand for galactose 1-phosphate uridyl transferase. And UDP galactose is converted to UDP glucose. This enzyme requires an enzyme epimerase. If any of these enzymes is defective, it will result into galactosemia. But the most common enzyme deficiency is galactose 1-phosphate uridyl transferase defect. This one here is the most common enzyme defect. So the classical in biochemistry means most common. So it is galactose 1-phosphate uridyl transferase defect. The presenting feature here will be vomiting, diarrhea, acute liver failure, like features, hyperbilirubinemia will be there, and there is a classical presentation, and that is congenital bilateral cataract. Then we have another question. Which enzyme deficiency will lead to the account for hemolytic anemia? The most common enzyme defect which results in hemolytic anemia is glucose 6-phosphate dehydrogenase. Let's see why. Now see, what happens is you have protons and you have oxygen in the RBC. The two can combine to form hydrogen peroxide. We must convert this hydrogen peroxide to water. For this we need 2 molecules of proton. These 2 protons are required. That 2 protons will come from 2 molecules of reduced glutathione. Now reduced glutathione will give this hydrogen and two molecules will join together by a disulfide linkage. Now it is known as oxidized glutathione. Now here we must convert this glutathione back to its reduced form. Now here we need NADPH. NADPH will give its protons, and those two protons will be converting this to reduced form. Of course, NADP will be formed. This NADPH comes from the enzyme G6PD. If there is deficiency of G6PD, there will be no NADPH, there will be no formation of reduced glutathione. Oxidized glutathione will keep on accumulating, hydrogen peroxide will keep on accumulating, and this will be converted to superoxide free radical. This will be converted to superoxide free radical, and more is superoxide free radical, more will be the chances of hemolytic anemia. So this is the problem with the G6PD deficiency. Just one second. So that is the problem with G6PD deficiency. Now, let's also see the other options. Now here glucose 6-phosphatase, glucose 6-phosphate dehydrogenase, glutathione reductase, glucokinase. The most common cause of hemolytic anemia under enzyme defect is glucose 6-phosphate dehydrogenase deficiency. Other things which can result in hemolytic anemia is, as far as enzyme defects is caused, pyruvate kinase deficiency, aldolase A deficiency, and PFK1 deficiency. These are the enzymes of glycolysis, and because the only source of energy in the RBC comes from glycolysis, this here is the another causes of lysis, because if there will be no energy, RBC will not be able to maintain its shape and it will lyse. Then another question, which of the following enzyme is thymine dependent and essential for glucose oxidation in brain? Answer is PDH. Now, let's see B1 dependent enzymes. Number 1, B1 is required for 3 oxidative decarboxylation reactions. These are those reactions which are made of, which have a similar mechanism of action. These enzymes are made of 3 different parts and they require 5 coenzymes: thymine, lipoic acid, coenzyme A, FAD and NAD. These 5 things are required by this enzyme. Now, these enzymes include PDH, alpha ketoglutarate dehydrogenase, and branch chain ketoacid dehydrogenase. These 3 enzymes are oxidative decarboxylation reactions. Number 2, we have transketolase. These 2 enzymes, basically one oxidative decarboxylation and another transketolase, these all enzymes require thiamine, and thiamine is also required for transketolase, and if you see, option A is transketolase. But is this enzyme essential for glucose oxidation via glycolysis? Transketolase is an enzyme of HMP shunt which is responsible for partial oxidation. Complete oxidation, that is conversion of glucose to carbon dioxide and water, that is brought about by the enzyme PDH. So the correct answer is PDH. Then another question. If there is any question regarding these questions or anything which you have studied under carbohydrate or anything in biochemistry which you are not able to understand, put it in the chat box. Which one of the following statements is true about TCA? It provides acetyl-CoA for the synthesis of fatty acid. It slows down when energy levels are low. It is the source of ATP in anaerobic tissue. It oxidizes acetyl-CoA obtained from fatty acid oxidation. Now, if you see, they are asking about TCA. It provides acetyl-CoA. No, it uses acetyl-CoA. It does not provide acetyl-CoA. It uses acetyl-CoA, combines with oxaloacetate, and it will undergo complete oxidation. It slows down when energy is low. Whenever energy is low, TCA should be high because TCA is the source of ATP. Now, it is the source of ATP, but it is the source of ATP in aerobic tissues, not in anaerobic tissue. The correct statement is it oxidizes acetyl-CoA from fatty acid oxidation. However, the statement is incomplete. Whether you are consuming fat, whether you are consuming carbohydrates, whether it is protein in the body, whether it is alcohol in the body, whether it is ketone bodies in the body, everything will be forming acetyl-CoA, and the ultimate fate of acetyl-CoA is to enter into TCA for complete oxidation. Then, which vacutainer is used for the blood glucose? Please note here that the red vacutainer is used for serum. Most of our things like LFT, KFT, lipid profile, everything is done on the serum. Gray vacutainer is used for the estimation of blood glucose. The EDTA vial will give you the blood. You can use it for ESR, for HbA1c, for CBC, because you're getting the whole blood here, for ABO-RH incompatibility, blood grouping, everything is done in the purple vacutainer or lavender vacutainer. Green can be used for the ABG or VBG assay. Green is used for arterial blood gas or venous blood gas assay. So the correct answer for blood glucose estimation is gray vacutainer. Which of the following is the major pathway in hepatocytes after 7 to 8 hours of fasting? Let's see a graph. This graph says that immediately after taking food will maintain the blood glucose for some time. After 8 hours, 2 to 4 hours, food will decline. That is, the food will no longer be able to maintain the blood glucose. After this process has declined, glycogen, liver glycogen will be the major source of energy. Some authors says it is 8 to 10 hours, others says it is 8 to 12, some says 8 to 14 hours depending upon the glycogen reserve. After this has declined, then will come gluconeogenesis. Gluconeogenesis will maintain the blood glucose for the next, let's say, 2 to 3 days. 2 to 3 days. And after this has declined, ketone bodies is the major source of energy. in our body. That means this, if we see particularly at 7 to 8 hours, we are somewhere here according to the question. And at 7 to 8 hours of fasting, the major source of energy comes from glycogenolysis, liver glycogen. Now I give you homework, you will find this out, why muscle glycogen never maintains the blood glucose. It's only liver glycogen maintaining the blood glucose, not the muscle glycogen. Why? Now, which of the following is not a reducing sugar? All monosaccharides, all disaccharides are reducing except sucrose, except lactulose, except trehalose. These will never give positive Benedict's test. In other words, these are the ones which give negative Benedicts. Negative Benedicts. So here the answer is sucrose. Which of the following is not a presentation in von Gerks? The most important presentation in von Gerks, number one, is fasting hypoglycemia. Because what is the problem in a patient who has von Gerks? This patient does not have the enzyme glucose 6-phosphatase. That means glucose 6-phosphate cannot be converted to free glucose. Now, if there is no free glucose, this patient will present with hypoglycemia. And when do we use glycogen? When we are fasting. So, fasting hypoglycemia, ketosis is seen because now cells will survive on the alternate source of the energy that will come from lipids. Hence, circulating lipids will increase, ketone bodies will increase, and there will be lactate formation because there is glucose 6-phosphate cannot be converted to glucose, but it can be converted to the lactate, and hyperuricemia is seen. Now, when glucose 6-phosphate increases, it can enter into the HMP shunt, and HMP shunt, when it is active, it will form lot of ribose. More ribose 5-phosphate is the starting substrate to form purines. More is purine, more is the chances of formation of uric acid. So the answer is hyperglycemia is not a feature, others are features. Rather, von Gerks will present with the low levels of glucose. Another question, a novice runner runs to a point where ATP utilization is faster than ATP can be regenerated by oxidative phosphorylation. Which of the following best explains ATP production in his skeletal muscles? So see, what this means is, the answer is A, regeneration of NAD. What this means is that glucose via series of reactions will form pyruvate. The pyruvate will then form lactate. This enzyme here is lactate dehydrogenase, and this enzyme will form NAD. It will consume NADH and form NAD. Now, everything here will be multiplied by 2 because glucose is 6 carbon, pyruvate is 3 carbon. So, everything will be multiplied by 2. Now, this 2 NAD is the same NAD which is utilized at the level of glyceraldehyde 3-phosphate dehydrogenase to form NADH. And this here, the last step, the significance of the last step is not to form lactate but to regenerate NAD, because in anaerobic tissues you need 2 ATP, and that can only come every time if you have a regular supply of NAD. What I mean to say is, a cell has a limited supply of NAD. NAD is not an abundant resource. You must regenerate it so that every time at least you get 2 ATP per glucose per anaerobic glycolysis. And for this you need NAD to be supplied continuously. So this last step of formation of lactate does not need lactate actually, but it needs NAD. Which of the following substance inhibits PFK1 enzyme? Now what happens is, let's see, glucose via series of reactions will first form fructose 6-phosphate, which will then form fructose 1,6-bisphosphate, which via series of reaction will form 2 molecules of pyruvate. These 2 molecules of pyruvate will enter inside the mitochondria to form acetyl-CoA by the enzyme pyruvate dehydrogenase. This acetyl-CoA will react with oxaloacetate to form citrate. Now when there is lot of this going on, there is a rate limiting enzyme of TCA known as isocitrate dehydrogenase. This enzyme is inhibited by NADH and it is inhibited by ATP. The citrate accumulates and this citrate will go outside. When there starts overflowing, that means you have lot of energy. When the citrate is overflowing a lot, that means there is lot of energy, and this citrate goes and inhibits this step, PFK1. It goes and inhibits the step PFK1. So citrate will go and inhibit the step PFK1. Now another question. Urine screening for an apparently healthy woman demonstrates positive Clean test. That means positive Benedict's test. However, blood glucose levels are within normal limits. More specific testing for urine glucose is negative. So the woman is positive Benedict's, but it is not due to glucose. It is due to which compound? This is due to fructose. Why fructose? I got that from the options, because when fructokinase is deficient, fructose will accumulate. When fructose 1-phosphate aldolase is deficient, then fructose 1-phosphate will accumulate. When the galactose 1-phosphate uridyl transferase is deficient, then galactose 1-phosphate will accumulate. And if lactase is deficient, then lactose will accumulate. Now, this can also result in positive Benedict's test. This can also result in positive Benedict's test, but as the question says, is unaware of any metabolic problems and leads a normal life. Lactase deficiency will cause bloating, abdominal pain, all those features. Fructose metabolism will not cause all those features. Hence, the answer is fructokinase deficiency, in which fructose will accumulate, essential fructose urea will occur, and that will give positive Benedict's test. Another question. A very ill infant is admitted to the hospital. Lab analysis reveals high serum concentration of lactate. In addition to taking the steps to correct acidosis, prescribes thiamine. Which enzyme uses thiamine? We discussed this earlier. Pyruvate dehydrogenase requires thiamine. Lipoic acid. It also requires coenzyme A, FAD, NAD. So these are 5 things required for PDH, and easy way to remember this is tender love care for Nancy. Those are the 5 things required for the enzyme PDH. Then all the cells show glucose uptake independent of insulin except. Now we divide the GLUT2 and GLUT4 amongst insulin dependent and insulin independent. I write 2 like this, and that helps me remember that this one will be insulin independent. That automatically makes GLUT4 insulin dependent. This one will require insulin. Now GLUT2 is present on liver. It is present in pancreas, small intestine and kidneys. Whenever this question is asked, it is asked in reference to GLUT2 and GLUT4. Now GLUT4 is present on heart, skeletal muscle and adipose tissue. So if you see here, out of the options given, GLUT2, pancreas is here, and hence that is the answer. Which of the following? Uptake independent of insulin except. So this one is except here. My mistake, I missed that except point. Yes, I missed that except point. So if we see, this one is independent. Now the problem is, whenever they ask this question, it is in reference to only GLUT2 and GLUT4. Now if you see here, heart, skeletal muscle and adipose tissue, out of this adipose tissue is given here. So here is adipose tissue, here is adipose tissue, hence that is the answer. So if the question would have asked glucose uptake independent, then my answer is pancreas. But if it is asked independent except, now my answer is adipose cell. I missed this except. Do not miss on this. Do not make the mistake I just did. Do not miss not, except, all, except. These terminologies are important. Now what is the order of draw of the blood samples in a vial? Whenever you take the sample from the patient, it first goes into the blue vacutainer. After the blue vacutainer, it should go into the red or the yellow vacutainer. Both are serum separators. Then it should go into the green vacutainer, then in the lavender vacutainer, then in the gray vacutainer. So if you see here, according to this blue, red, lavender and gray, the correct order of draw is blue, red, lavender, gray. B, option B. So the correct order is this. So this here is the way we have written. Now whether you call it red, yellow or golden, it's one and the same, because all of them are serum separators. So it is one and the same thing. That is why I have written it same. And that is why here it is mentioned like this. What is the enzyme deficient in Pompey's? Pompey's is a disorder in which the enzyme lysosomal acid maltase is deficient. Alpha-1416 glucosidase is deficient. The Pompey's is the only lysosomal disorder of the glycogen storage diseases. In this disorder, the glycogen-like granule will accumulate in the lysosomes, and this is a poor prognosis disease. These glycogen, after its half-life is over, it should be degraded, but here we are not able to degrade it. So it will keep on accumulating, and these children, they usually die because of heart failure. Then properties of HMP are all except. It produces NADPH, yes. It is producing ribose, yes. Is it utilizing ribose? Yes, you can say both, because it produces also, and in the next step it utilizes also ribose. Now, one of the ways by which the ribose produced is used is also known as DNA synthesis or RNA synthesis. But you will never get acetyl-CoA. Now here what happens is you start with 6-glucose. You will form 6 carbon dioxide, 6 water, and you will get here in the end, you will get here 5 glucose. So only 1 glucose is oxidized at a time. So you start with 6, only 1 is oxidized. This HMP shunt is hence partial oxidation, resulting in the formation of carbon dioxide and water only from 1 glucose. Rest 5 glucose you will get as it is. So this HMP shunt is an alternate way. I know it is not very effective, unlike glycolysis, where if we start with 6 glucose, you will get 6 carbon dioxide and depending upon 36 carbon dioxide and 36 water, depending upon the number of carbon atoms per glucose, that is 6. So 6 into 6, that will become 36 plus 36 each of carbon dioxide and water. Now, which of the following enzyme of glucose metabolism participate in gluconeogenesis? Now, let's see. PepsiK is the enzyme of gluconeogenesis. Glucose 6-phosphatase is an enzyme. Hexokinase belongs only to glycolysis. Pyruvate carboxylase is an enzyme of gluconeogenesis. Now, here they have written glucose 6-phosphate. I am not sure whether it is glucose 6-phosphatase or not. But if it is this case, so we will consider it as glucose 6-phosphatase, which is same as option B. So this here is, or because it is not clearly written here, it is not clearly mentioned here, either if it is considered to be glucose 6-phosphatase, then both B and E is the same option. So here we will ignore this option, we will cancel this option. So my correct answer should be A, B and D. So do we have an option like A, B and D? Yes, there is an option. So these are the enzymes that participate in gluconeogenesis. This is based on an important fact that there are 3 enzymes of glycolysis. Number 1, glucokinase hexokinase. Number 2, PFK1. And number 3, pyruvate kinase. These 3 enzymes are reversed by gluconeogenesis 4 steps: PC, PepsiK, the glucose 6-phosphatase, fructose 1,6-bisphosphatase, which we have already written on a previous question. Rest all the enzymes of glycolysis are reversible and common to both glycolysis and gluconeogenesis. Which enzyme defect is McArdle's? It is muscle phosphorylase. And if somebody does not have muscle glycogen phosphorylase, when the muscle is exercising, energy will not be produced. Hence, the most important feature is exercise intolerance. The most important feature will be exercise intolerance. Under glycogen storage diseases, you have to know all the names of type 1, 2, 3, 4, 5, 6 and the enzyme defects. You can even get a match the following type of question in glycogen storage diseases. Most important topic under carbohydrate is glycogenolysis, glycogen storage diseases. Now, why glucose 6-phosphatase cytoplasm is not acted upon by glucose 6-phosphatase as soon as it is formed? Now, this is due to the enzyme is present, glucose 6-phosphatase is present in the cytosol. Let's try and understand this. The question is that glucose is converted to glucose 6-phosphate. Now, glucose 6-phosphate is converted to glucose. Here we need an enzyme glucokinase. Here this enzyme is glucose 6-phosphatase. Now why glucose 6-phosphatase does not act upon the glucose 6-phosphate formed from this glucose? Why it does not convert it back? How is this futile cycle prevented? How this conversion to glucose and then to this is prevented? What nature did was, this glucose 6-phosphatase is inside the endoplasmic reticulum. It is not a cytosolic enzyme. So whenever glucose 6-phosphate is in excess, it has to enter inside the endoplasmic reticulum to be converted to glucose. To be converted. This step will not occur outside. This has to be inside. Then, which of the following is not an intermediate of TCA? Citrate is an intermediate. Succinyl-CoA is an intermediate. Alpha-ketoglutarate is an intermediate. Propionyl-CoA is not. I use a mnemonic to remember the intermediates of TCA. Citrate is Krebs starting substrate for making oxaloacetate. Now if you compare this, open your book where you have written seen TCA, open your book and compare this. Citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate. In that order. Substrate used by RBC in the fasting state. Whether you are fasting, whether you are well fed does not matter to the RBC. It only uses one source of energy and that is glucose. RBC always uses only one source of energy and that is glucose. RBC does not have mitochondria. It cannot use ketone bodies, fatty acids and amino acids. So with this we have covered your... there is one more question. Lactic acidosis in thymine deficiency is due to which enzyme defect? It is due to PDH. This was a similar question before. So with this we have covered your few questions. One last question still is left. The enzyme activated with low insulin to glucagon ratio. That means there is high glucagon and this high glucagon will activate gluconeogenesis. And the key enzyme for gluconeogenesis, I told you 4 enzymes. Glucose 6-phosphatase, fructose 1,6-bisphosphatase, pyruvate carboxylase, PepsiK. Out of which here we have glucose 6-phosphatase. Please note, insulin promotes glycolysis. Glucagon promotes gluconeogenesis. I wish you all the best. Keep studying. Make sure that you revise the carbohydrate metabolism thoroughly. Right? Now you will get some more insight into the topic. Thank you so much. Keep studying.